Good morning, everybody. You are the hale and the hearty, those who, even though they went to club night, are here at 8 a.m., so thank you so much. Let's see. Slides. Okay. I'd like to introduce myself. I'm Marie Banich from the University of Colorado at Boulder and organized this symposium on executive function and brain connectivity. I'd like to thank the other speakers for joining me this morning. So what I'd like to do as an introduction to the symposium is just give a brief overview, sort of what is executive function. There are lots of different theoretical frameworks that people have used, but generally it's a set of abilities that allow for goal-oriented behavior, and it really is an umbrella term that covers many different aspects of behavior, from setting goals to sequencing actions towards goals, overcoming distraction that would derail the goals, switching between tasks, and a myriad of other aspects of goal-directed behavior. The reason why we organized this symposium is that if you think of most of, or sort of the importance of executive function, it's disrupted in many populations. So it's a higher level cognitive function, it's disrupted in psychiatric disorders and neurological disorders. Hence, there's a widespread interest in its understanding its neural underpinnings. With regard to its neural underpinnings, as you all probably know, it relies preferentially on frontoparietal regions, is this laser pointer working? Frontoparietal regions with added involvement of other regions. What I've shown here is a reverse inference map from neurosynth using the term executive. Now, importantly, most theories of executive function suggest that what this neural system does is to modulate the processing of more distant brain regions. So I'm going to take one of my favorite tasks here, the Stroop task, and essentially models of executive function argue, for example, that what this frontal parietal network does is to, for example, in the case of the Stroop task, upregulate brain regions that are involved in color identification and or downregulate those regions that are involved in reading. So obviously, to exert this modulatory influence, it requires brain connectivity. And hence, this is sort of the rationale for our symposium today, because part and parcel of theoretical conceptions of executive function is this notion that brain connectivity must play an underlying role. Okay, so the outline of our symposium today, in the first talk I'll be talking about individual differences in brain organization, relatively static aspects of brain organization as it relates to executive function, EF, and brain connectivity. I will be followed by Lucina Uddin, who will be looking at more dynamic aspects of brain function as they apply to executive function. Bruce Morton will then follow up and give us perspective on these dynamics as they relate to developmental issues. And then Nathan will be giving the final talk, looking at the interaction between the executive function network and the default network as it helps in goal-directed cognition. So that's our outline for today. Okay, so without further ado, we'll get started. My goal today is to talk about individual differences in anatomical and functional aspects of brain connectivity as they influence components of executive function. I'd like to thank all my collaborators, both folks in my lab and faculty members with whom I've worked closely on this work, as well as my funding sources. So today what I'm going to try to do in my talk is to contrast and look for evidence of two different conceptual models of how executive function might be organized in the brain to subserve higher levels of executive function. So I'm going to contrast these two models, and it's a relatively simplistic contrast, but I think you'll get the idea. So one idea is that was shown on the left here is it could be that folks with high executive function have a souped-up, really great frontal parietal network. Okay, so that's kind of model number one, that this frontal parietal network is just supercharged, that they have better utilization of this core executive function network, and perhaps along with that, they might have more centralized control. It might be that the dorsolateral prefrontal cortex is the buzzing hub of activity for folks who have very good executive function. I'd like to contrast that today with a model that says, no, actually those folks with high executive function are basically able to bring in more brain resources, that they have more distributed control, that essentially their network expands beyond the core executive function network. They have this expanded or extended network, and perhaps also that the control their control in terms of executive control is more distributed. So I would like to talk to you about how I'm going to, or how we in our lab investigate this. One of the issues is that we use a particular model of executive function. So in terms of the psychological structure of executive function that we're going to use, it's a model that's been put forth by Miyake and Friedman, the unity and diversity model. And they have argued that across a variety of executive function tasks, there is a common basically measure, common EF. So much as we have the concept of G for general intelligence, in this model, the unity aspect of the model is considered common EF, this factor, this latent factor that all EF tasks rely on. And they have argued that the underlying psychological ability is the ability to maintain task goals online. So those are required for all executive function tasks. And that in addition to this unifying factor, that there are at least two other factors that are important in terms of, or latent factors supporting executive function, which is the diversity in the model, a shifting specific factor, that is the ability to shift between goals or tasks, and an updating specific factor, which is the ability to update working memory in service of goal-directed behavior. We use this model because it is basically designed to explain individual differences in executive function, and it has basically proved to be a very useful model from a number of different perspectives. What I'm going to talk about today are some multimodal investigations that have occurred in our lab. So we have looked at the relationship of these latent EF factors in terms of brain anatomy, in terms of resting state networks, intrinsic connectivity networks, which are rather stable and show coherent individual differences, and also resting state data in terms of the issue of graph theory. In particular, I will be comparing how nodes act as integrators. Do they connect disparate regions? Different measures of that. Are they a hub? Do they link to various different modules in the brain? And also looking at in part at what are the segregation characteristics of these nodes. So I'm going to talk about 3 studies, one at each of these levels. Just to give you an overview, the subjects' population size ranged from 70 to 90 individuals. These were individuals in their teens or relatively late teens, early 20s, relatively tight age range from about 19 to 21, 22. So what did we find? So for common EF, essentially what we found was that regions in the ventromedial prefrontal cortex predicted essentially performance on common EF. What we found is actually reduced gray matter in this region, reduced local gyrification index predicted better EF. So notice that this region, anatomically, this predictive, is not sitting within the core frontal parietal network. Okay? For shifting specific factor, we found those individuals with higher EF showed less gray matter volume and a lower local gyrification index in this region of right VLPFC. Okay. Once again, note that this is not a classic frontal parietal control network region. And then for updating specific EF, we found that those individuals who showed better updating specific EF showed decreased gray matter in a dorsolateral prefrontal region and also showed a reduced local gyrification index. Okay? So we're seeing clear anatomical variation with each of these different EF factors. Now, if we move on into intrinsic connectivity networks, this is what we find. So let me explain what these graphs look like. What I've plotted in the warm colors are essentially the intrinsic connectivity network as derived from resting state data. And then we did dual regression, which is looking at the loadings of individuals with regard to those networks. And what we look at then is how are those networks varying vis-a-vis levels of executive function. So what we found for common EF is that those folks who have have high EF, if you look at their frontal parietal network, they show increased amplitude in the cerebellum. Okay, so their frontal parietal network is showing increased amplitude in the cerebellum. And we also find for the ventral attention network that we see expansion to this more frontopolar region. For shifting specific EF, we find those individuals with higher shifting specific EF show expansion of the dorsal attention network to parietal regions, and we didn't find anything for updating. So notice once again that what we're seeing is expansion of this network rather than an increased, except in this case, an increased amplitude within the network. And then in our third study, this is a busy slide, but there are a couple take-home messages that I think you'll see. What we did was we looked at graph theory metrics. Once again, a resting state, so the brain's divided into 285 nodes, and we're looking at how the communication characteristics of those nodes vary with executive function. So two take-home messages from this slide that I think you'll be able to pick out. What I've done is I've put a star around those nodes that fall within the frontal parietal network. Any node that's on this graph is a node that we found was predictive, that its connectivity characteristics were predictive of EF. So two things to note. First of all, I'd like you to notice that there are a lot of little nodes on this slide that don't fall within the frontal parietal network. Okay? So first of all, first take-home message from this slide is that there are a number of nodes outside of the traditional frontal parietal network whose connectivity characteristics predict levels of executive function. Then the other thing I've done for those nodes within the frontal parietal network, what I've done is either put a red circle around them or starburst or a blue one. Okay? And essentially what I'm showing you, if it's red, it means that if you had high EF, that particular node was more hub-like, it did more integration, versus if it's got a blue starburst around it, less integration was at that hub was associated with higher EF. And I think as you can see pretty clearly, there are more blue stars than red stars. Okay? So we're finding that actually higher EF is associated with nodal characteristics that are showing less integration or less hubness. Okay. So this is just a summary slide where what I've done is I've taken the three studies I've just talked about and I've sort of put a little symbol to indicate whether the results were more consistent with the great centralized control model or were more consistent with a model of more diffuse organization, greater connectivity, greater diffuse expanded network with individuals with higher EF. And I think you can see from this simplistic little diagram that the bulk of the evidence suggests that those individuals who are higher in EF have actually a more diffuse network, not a more focally organized network. Okay, in the minute I have left, I just wanted to show you an exploratory question we are in the process of examining. One of the things we wanted to do, since these data were all on the same people, is to try to see whether these different levels of information, the anatomical features, the intrinsic networks, the network organization, are they reflecting one underlying biological factor, or are they each contributing separately to predict executive function? So I won't go into the modeling and the bootstrapping and all the other things we've done, but just to give you an overview, the main take-home from this slide is that we are finding that anatomy and the intrinsic connectivity networks and the graph theory each seem to be providing different variants in a predictive manner. So the variants I have on the left there are basically sort of average variants for bootstrapped models. I'm sure they're an overestimate, but if you took random factors, you only account for basically about 20% of the variance. So our initial data suggests that these different levels of brain organization are each bringing unique variants. So then just to finish up, our summary, individual differences in EF are associated with organization of intrinsic brain networks, information flow through nodes, and structural characteristics. Also notice that the specific pattern differs for each EF component, suggesting they are indeed psychologically separable. In general, higher EF is associated with a more diffuse and centralized brain organization, and going forward, we're working on looking at these issues in a similar way in a larger cohort of individuals who are somewhat older. Our initial results suggest a similar pattern, but we'll also be looking for maturational changes. Thank you very much. Okay. Lucina?
Okay. Can everyone see me or hear me? All right. I can't see any of you. So thank you very much for coming this morning. Thanks very much again to Marie for organizing this symposium. To the OHBM program committee, I'm 80% honored and only 20% annoyed to be here at this coveted spot at 8 a.m. after club night, but thank you all anyway. So basically, in keeping with this theme of the symposium, I just want to start out with some of the reasons why we're interested in executive functions. And this is work by Dina Dajani, who's a grad student in the lab. And if you are interested, you can see the poster, which is probably still up for the rest of today. But what she's done is started to look at this question of how executive functions are very important in neurodevelopmental disorders for predicting outcomes in various domains. And the long story short is that if you look at a mixed group of children with autism, attention deficit disorder, and also typically developing children, you can actually stratify these children based on their executive function profiles, such that you get some with sort of above average executive function abilities, a sort of a middle group, and a below average performing group. And this is done with a latent profile analysis using many parent report and lab-based measures of executive function. I should mention this is done in collaboration with Stuart Mostovsky's group at Johns Hopkins. And the poster describes some of the brain network differences that also differentiate the executive functioning levels, and I won't get into that today. But I wanted to highlight that you see these sort of three distinct classes of individuals with differing executive function profiles, and typically developing kids tend to fall in the above average group, whereas kids with autism, kids with ADHD, and kids with both autism and ADHD tend to be sort of the most impaired in their abilities. And what this is interesting because this actually predicts a host of behavioral problems, anxiety, depression, aggression and many other issues that these kids experience. So having a sort of a low executive function ability tends to predispose these kids with developmental disorders to be even worse off in several domains. So we're interested in figuring out sort of why this is the case. And yeah, just because it's early, I wrote everything out. These results illustrate how development of executive function abilities impact a range of behaviors in some of the most prevalent neurodevelopmental disorders. Now, we know from the nice introduction we had early on today that we tend to think of these frontoparietal regions of the brain being very important for subserving the umbrella term of executive functions, with maybe a few of these nodes relatively specialized for specific processes like task switching, working memory, attention, and various other things that go under the umbrella of executive functions. But as a field, I think we're also moving away from the sort of node-based descriptions of cognition to more network-based descriptions. And I particularly like to go back to the early work from Randy McIntosh and colleagues, where he describes this idea of neural context. And like every other cognitive function, I think it makes sense to think of executive function in the same way. So from this 2004 paper, I just have the quote that the functional relevance of a brain area depends on the status of other connected areas. So the context within which a brain region is operating is, of course, very important for understanding its function. So in this figure from that paper, you can see these 4 nodes that are connected in a particular way may behave in a different fashion just as a function of the patterns of connectivity with its connection partners, whether they're active and the way that they're active might influence the function of those particular regions. So I think this is always a nice thing to remember when we're looking at things that we think are important for a specific function. So with that in mind, we're starting to think about brain dynamics and sort of whole brain connectivity contexts that might be uniquely predictive of executive function abilities. So this is work that Jason Nomi, a postdoctoral fellow in the lab, presented yesterday in the oral session. I believe the poster might still be up, 4084. And what he's done here is take some of this really nice Human Connectome Project data that's been made publicly available, and a lot of good things about this data set. There's about, here we took about 200 of the subjects, neurotypical adults, and the team has already provided the independent component maps, the time courses. A lot of processing has already been done to this data. And you can see, you can get a really fine parcellation of the brain with many regions that we're familiar with. And so we've just grouped them here into some networks to facilitate the interpretation of the horrible correlation matrix that I'm going to show you next. But as many of you know, with dynamic functional network connectivity, in particular this sliding window method that's been utilized by Vince Calhoun's group in the GIFT software, you basically can start with an independent component analysis to parcellate your brain into various regions. And then instead of sort of taking the every region by every region and looking at the correlation across the duration of the scan, you're doing sliding windows according to whatever window length you decide on. And you end up with many, 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 many correlation matrices describing the dynamics of connections across the course of the scan. In this case, the participants had 1 hour of resting state fMRI data, so really many, 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 many correlation matrices that then can be clustered into groups, and here we decided on a cluster solution of 5. And these sort of 5 distinct brain states describe the patterns of dynamics of connectivity across this entire hour that each of these participants willingly stayed in the scanner. And so I also stole this slide from Jason's talk yesterday, so we can thank him for that. So basically, you can come up with brain states, and you can also look at the trajectory through these different brain states. So for example, an individual might be in state 1 and in state 1 and then transition to state 2, so you can sort of quantify that. You can look at the frequency of occurrence of states, so some states will show up more often than others. And all of these things, dwell time is another measure, which is basically how long does a brain stay in a particular one of these brain states. So many indices that can be quantified, and for our purposes, we can actually try to correlate these with executive function abilities, which is where I'm going with this. And in the HCP data, of course, you have this really nice behavioral characterization. You have all these executive functions that we can look at and try to see if there's any relationship with the brain state metrics that I've shown you here. So what we found here is at the top row, we've got the 5 brain states ordered by frequency of occurrence. So, state 1 occurs about 40% of the time, with state 5 about 10% of the time. And already just by ordering them in this way, you can see there's something interesting about the way the brain tends to be. And state 1, which is the most frequently occurring, is sort of a more attenuated pattern of correlations that are, centered around 0, not too extreme. Whereas state 5 is the kind of like really strong positive correlations between regions and really strong negative correlations, more dispersed, more extreme patterns. And that's what I have here. Oh, yeah, mean correlations. And the histogram is just showing you the strength of the connections, across these states. And the third row is just showing you the standard deviations. So, state 1, there's kind of a greater range of possibilities of the connection strengths. And the reason I'm showing you this is to help us to try to understand what the sort of significance is of a brain that happens to be the propensity to be in one state versus the other, and what we can think that means in terms of cognitive processing. So at the bottom here, if we can see that, basically what we found is that individuals who tend to be or have a frequency of occurrence of state 1 that's greater tend to be better on the list sort measure. What is that? Yeah, you can barely see here. So I think the next slide will actually summarize this better. Yeah. So if you look at each of these sort of measures from the HCP data set, there's a few relationships, and most of them were actually only the relationships that we found were with frequency of occurrence of particular brain states. So if you look at both the card sort and the list sort, the individuals who spent sort of more time in state 2 and less in state 3, as well as individuals who spent, the frequency of occurrence of state 1 was greater than state 5. Those were the individuals that showed the best performance on these measures of executive function. So here's the really long-winded way of saying that the brains of individuals who perform better on measures of cognitive flexibility and working memory are characterized by the tendency to occupy more frequently occurring brain states. And those were the states, if you remember, that present with correlation distributions that are around 0, hovering around 0, and have larger variability or greater standard deviation, rather than the less frequently occurring states that tend to have the more dispersed correlation distributions and lower variability. So, do I have a take-home message? Yeah. These results highlight the potential for dynamic functional connectivity approaches to reveal relationships between dynamic brain states and executive function abilities. And, of course, that poster is still up if you want to hear more details about that from Jason today. And finally, this is the part of the talk where if you're starting to fall asleep, I hope this will bring you back. So here we're actually testing a theory about network interactions, and we've been working with this model for a while where we think that there may be a particular significance to a set of regions we've called the salience network, in that oftentimes you can find that activity in these regions sort of precedes activity in the central executive and default mode network, and you can use signals from the insula and other regions to sort of predict what's going to happen in the other two. So the idea is that perhaps the salience network and insula in particular is involved in kind of dynamic switching between two of these other large-scale brain networks. And with this in mind, a visiting grad student from Berlin, Rosa Stemke, came to our lab and set up to test an idea that something about these relationships between these networks and their dynamics might have an impact on individual differences in executive control and self-control in particular. And this poster is gone. So here's the task. All you have to do is press one button if you see the letter E and press a different button if you see the letter F. So this should be a kind of easy task to do. Let's see if we can do this. Oh, it's not a video. I'll click through it. Okay. So basically, that's where you're supposed to focus. Keep focusing. Should be an E or an F. And keep in mind that for these participants, eye tracking data were collected. Okay. So most participants are looking over there because I told you to. But sometimes a distracting image appears, And then participants tend to not look where you're supposed to be looking, and they might miss that E or that F that was supposed to be the thing you're monitoring for. So people vary in their ability to stay focused on the E or F in the face of these very, distracting images. And as you can tell from the eye gaze patterns, many people end up really missing that target, and you can imagine there's a lot of individual differences in the ability to really do this thing. And as you can see from the top here, in terms of reaction time, in terms of error rates, in terms of the eye gaze, all of these things are affected by this erotic distractor. So most people do worse and look away from the target when faced with such temptation. This was done in Germany. I really had nothing to do with this data collection, but I'm very grateful that we were able to explore it. So these are just some of the eye tracking. These are all just behavioral tests done outside the scanner, and I think it was done on about 100 college students. Now, the beautiful part for us was that the next day they came back to the scanner, and they got resting state data, 5 minutes of rest, and we ran the dynamic functional connectivity analysis that I just described, except this time we really just focused on the salience network. So the idea is there something about the dynamics of that salience network that is different in the individuals who are really good at this task versus those who are not so great at it? And the other hypothesis you might also have had is that maybe the cognitive control system or executive control system is actually more linked to individual differences on this task. So we sort of pitted those two ideas against each other. I can wrap up real quick. So basically, just looking at salience network states, we have 5 states that come about. And I want to focus in particular on this state 3, which occurs about 15% of the time. And if you look at it closely, you'll see that the salience network seems to be negatively correlated with the visual system. That blue chunk in the middle is the visual network. And in this state, salience and visual regions tend to be sort of not talking to each other. And it turns out the only significant relationship with behavior was that those individuals who spent the most time in state 3, or I guess it was frequency of occurrence of state 3, are those that were better able to resist the tempting distractors. The better performers were those whose brains actually have a propensity to stay in a state where their salience networks and visual networks are not really talking to each other as much. So they have somehow are able to resist all that temptation. So these results highlight the unique ways in which salience network dynamics are related to individual differences in self-control. And finally, just to wrap up in 10 seconds, I just want to end with this quote from 2002, that the functions of a cortical area are determined by its extrinsic connections and its intrinsic properties. But as we're starting to realize this also, the factor of time is also very relevant for understanding some of these brain behavior relationships. And I want to thank the lab, Jason Omi, Dina Dajani, who did the work that I showed today, Rosa Stemke in Berlin, Vince Calhoun's group, whose method we've used, Stuart Mostovsky at Johns Hopkins, NIH. And if you're interested in this, I'm going to use this last 5 seconds to plug the book I have that's coming out in September. So please check it out. And this is the lab. Thank you very much.
Okay. Well, thank you all very much for coming. It's a pleasure for me to be here. Thank you, Marie, for the invitation to participate in what's really a very interesting symposium. So across development, then, we see widespread changes in the speed and complexity of behavior, and perhaps this is most pronounced in the higher order domains of cognition, such as executive functioning. And this is nicely illustrated, for instance, if you look at age-related changes in performance in stimulus-response compatibility tasks, where subjects are presented with a target task. In this case, in this figure I've shown here, the target task is to identify which of two numbers is numerically larger, and mixed in with the stimulus is a relevant feature, in this case, the physical size of the stimulus. So on compatible trials, the target task is in some sense facilitated by this relevant information insofar as the numerically larger stimulus is also physically larger. Whereas on incompatible trials, the irrelevant feature is incompatible with the target task. So the numerically larger stimulus is physically smaller, and this introduces a small cost in performance, which is measurable by the interference effect and reflected both in an elevated rate of errors and also prolonged response time. Now, from a developmental standpoint, we look in the bottom panels on the left, you see that across age, shown on the x-axis, then you see a gradual attenuation of the magnitude of the interference effect with increasing age. And you see the same thing then in the magnitude of the interference effect measured with response time. Again, with increasing age, there's a diminution in the magnitude of the interference effect with increasing age. So these age-related changes then in executive functioning have long been thought to be related in some way to functional changes within a broad network that we might describe as the cognitive control network. It's a broad network encompassing both, or including lateral prefrontal cortex, cingulate cortex, anterior insular cortex, regions of premotor cortex, parietal cortex. And they're thought to be a network insofar as these regions tend to co-activate in response to the administration of a cognitive control task. And if you look at the correlations between these regions while participants are at rest, you find that the time courses extracted from these areas tend to be positively correlated with one another. Now, to date, developmental researchers have really looked at changes, age-related changes in the spatial features of this network in an effort to try and understand changes in executive functioning over developmental time. And this is best exemplified by a graph theoretical approach forwarded in particular by Damien Fair and colleagues in the St. Louis group. And in this approach, of course, participants are scanned while they're at rest, and then on the basis of the correlation profile from amongst areas that comprise the cognitive control network, then one then constructs these graphical characterizations of network organization. And the assumption underlying this approach, of course, is that the spatial features of these networks remain invariant for the duration of the measurement period, and that they're an intrinsic property of the network. In other words, they don't really vary as a function of what it is that the subject is doing as the data are acquired. Therefore, the characterization of age-related changes then are largely based on these topological differences in network organization across developmental time displayed here. So this approach obviously has had a vast influence on the field and certainly is very important, if for no other reason than to call attention to the profound changes in functional brain organization that occur with development. But as Matthew Hutchison and I call attention to in a recent critical review published in Developmental Cognitive Neuroscience, the graph theoretical approach also has important shortcomings. And the critical one, in our view, is the fact that the correlations upon which any of these graphical characterizations of network organization are based are unreliable for durations, that are typically what one uses when administering resting state paradigms. And so if these correlations are unreliable, then one has to admit that the assumption that these spatial features are invariant during the measurement period then is difficult to support. So the spatial features themselves are then subject to variation over time scales that we use to measure network organization. Moreover, Lucina's beautiful findings notwithstanding, these graphical characterizations are also difficult to link to function, at least directly in terms of what a participant is doing while the data are being acquired. So for this reason, Matthew Hutchison and I motivate or advocate for a dynamic approach to the study of age-related changes in the cognitive control network, an account that allows for the fact that these spatial features will vary over time. And we think that this is particularly important owing to the fact that, according to existing theory, the key constraints that govern the emergence of these dynamics are things, for instance, like the path length, the connecting nodes in the network, the rate at which signals travel along the connections between nodes in the network, and the degree of noise that's carried by these signals as they migrate through the brain as well. And these constraints that govern the emergent dynamics of the system are subject to developmental change owing to changes in the size of the brain, myelination of the axonal pathways that convey signals in the brain, and as well as changes in neurotransmitter levels and receptor levels that also occur with development. So one should expect then not only that the system is dynamic, but the nature of these dynamics should be subject to developmental change. So currently our empirical program really right now is to, first of all, to identify whether the method is reliable and valid in the study of developmental variability, and then to go one step further and to begin to link some of the products of this dynamic approach then to functional changes in the instantiation of control. So one paper that Matthew and I published recently in the Journal of Neuroscience investigated 51 participants ranging in age from 9 to 32 years. And each of these participants then were administered a standard resting state paradigm. And in addition, we administered this size congruency or response compatibility task, as I described earlier. The data then were subjected to an ICA-based cortical parcellation, which gave us 56 time courses. Then that, as Lucina described very nicely, we then subjected to a standard sliding window approach, which generated a large number of these sort of time-resolved functional connectivity matrices that then are concatenated into a data matrix. And then using a multivariate decomposition, in this case a k-means clustering algorithm, then we were able to examine both spatial features, in other words, spatial features in the data as well as temporal features in the data. And by the spatial features, I mean these are features, these are continuously recurring spatial patterns that are evident in the data. And then the temporal features, which is shown in the bottom panel on the left, basically for each individual participant, then you get what's called a state vector, which just shows on the x-axis the various windows that were acquired from subjects either while they were at rest or at task. And it shows you how the clustering algorithm then classified each of those windows. And then you can look at that vector as Lucina described, and you can derive a number of dynamic metrics from that, including the number of transitions that participants make, and how long they remain in a particular state, and you can compare those across task and rest for participants of different ages. So, taking a look at the results, I just want to focus first on the spatial features that were revealed by this clustering algorithm. And the first thing to take note of is that the network organization that is observed using the traditional mean FC approach to understanding functional connectivity, that the borders between those networks that are evident in the leftmost matrix there, this mean FC matrix, where you see this kind of clustering along the diagonal, that each of these dynamic states then shows very pronounced topological migration away from these canonical or traditional network boundaries. So, for instance, if you look at state A, in the upper left corner of that matrix, the somatomotor network has largely dissolved, but the default mode network is profoundly intact. Whereas if you switch to state C, for instance, again, looking at the upper left corner of the matrix, you can see that the somatomotor network now is very tightly connected to itself, whereas the default mode network now has largely dissolved. And there's also very, very interesting and complex off-diagonal changes in the topology of these matrices. So the simple take-home message is that the structural features of any of these networks, let alone the cognitive control network that we see when we use the traditional approaches, disappears when we begin to look at the spatial features as revealed in a dynamic analysis. Importantly, though, these spatial features are reliable and consistent across participants of different ages. So to look at this in particular data set, what we did is we looked at all of the exemplars from adults that were, for example, classified as an instance of state A. And we compared those with exemplars that had been acquired from children and had been classified by the k-means algorithm as instances of state A. And each of those then exemplars was correlated with the centroid of that category. And you can see for all 12 of the centroids that were extracted from the data, the exemplars for the adults and the children are quite similar, right? So this is important because it I think, highlights the fact that across development then, at least in this dynamic analysis, the spatial differences disappear then when you look at age variability. So you might ask, well, where are the age-related differences? And the age-related differences in this analysis came out in our examination of the temporal features. So and I don't have time to go into detail here, but let me emphasize two metrics that we derived from analysis of these state vectors. And one was just the number of transitions that were made when subjects were at rest and at task. And in fact, the most interesting associations was observed during the administration of the task. So in this scatter plot, age is on the x-axis, the number of transitions is on the y-axis. The data for the resting scan is in blue, and the data for the task scan is in red. And what you can see is that there's no association between the number of transitions and age during the rest scan. However, during the administration of the task, we see that older participants are showing more transitions than our younger participants. This is reflected in another metric that can be derived from an analysis of these state vectors, namely the time between transitions. And here, again, there's no association between age and the time between transitions when we look at the resting data. However, when we look at the task data, we see that there's a negative correlation between age and time between transitions. So the take-home message here is that across age, we see changes in executive functioning measures such as magnitude of interference effects, which have been attributed to changes in these spatial features. But a dynamic analysis would suggest actually that this cognitive control network that we've really been focusing on for a long time has actually no fixed topology, but it dynamically varies over relatively short timescales. And importantly, age-related differences now are more actually evident in the dynamic or time-based metrics than in metrics that are derived or from a focus on the spatial features. And the final thing that was kind of interesting in this, in trying to build a functional story, is that in adults, we see an expansion of the state repertoire as cognitive control demands increase. This is just based on an examination of the task data relative to the rest data for the adults, where there was, again, an expansion of the state repertoire during task relative to rest. But I admit that this is not an altogether compelling illustration of the importance of the state repertoire for function, again, because there are many complexities and challenges associated with comparing task data and rest data. So we wanted to finesse this in a little bit more detail. And in order to do this, we took advantage of the fact that in this size congruency task, you can change. My counter is coming down here. Scares me. We can parametrically vary the magnitude of the interference effect by just changing the saturation of a condition with the compatible trials. So, for instance, in a 25% compatible condition, only 25% of the trials are compatible. Most of them are these incompatible trials. In a 50% compatible condition, half are compatible, half are incompatible. And in a 75% compatible condition, most of the trials now are the compatible type, and once every so often you get one of these hard trials which kind of jars you back into the task. And what you see when you parametrically vary the saturation of the condition with compatible trials in this way is that you get a nice parametric manipulation of the magnitude of the interference effect. This is referred to as the Croton effect. So we took advantage of this effect and focused our analysis on 20 adults and just compared the dynamics in the 25%, 50%, and 75% condition using our dynamic pipeline. The one difference that we made here in this analysis is that we applied a different multivariate procedure to decomposing the concatenated data matrix, which I would be happy to talk to you about afterwards. But this allowed us to come up with a more nuanced estimate of repertoire complexity than is possible with the k-means. So as predicted, we see this very nice parametric manipulation or parametric change in the magnitude of the interference effect across these conditions, with the magnitude of that getting increasingly large as the saturation of the condition with compatible trials increased. And what's interesting to us, and which we'll continue to pursue in more detail, is that we see, again, this very nice parametric increase in the size of the repertoire, the dynamic repertoire, with increases in the saturation of the condition with compatible trials. So we're trying to, using these methods then to build a story which will allow us then to extract measures of dynamics from the data and data that are acquired as participants are performing cognitive control tests that parametrically vary in terms of their demands on cognitive control. So stay tuned. In any case, for now, what I would just like to say is that age-related changes, at least from a dynamic conception, are more apparent in the temporal features of the cognitive control network, sort of how these spatial features change over time, than in the spatial features themselves. And this is an important departure from the traditional understanding of developmental change that we have based on a graph theoretical approach to understanding cognitive control network development. And links between temporal features and cognitive function, I admit, are still tenuous, but there's some evidence to suggest the importance of an expanded repertoire of states. And we, as I said, will continue to pursue this in the future, both in our work with adults and with populations that vary in age. So with that, I would like to acknowledge the assistance of a number of people, my collaborator, Matthew Hutchison. I'd like to thank Elena Allen and Tom Eichela for their assistance in orienting me both to ICA and to dynamic functional connectivity analysis. Vince Calhoun's group for ongoing assistance with coding and various other things, and folks in my lab and people in my department, Daniel Lewis and Jim Neufeld, for just talking math and code, and to my funding agencies that allow me to be here to talk to you all today. So thank you very much for coming, and yeah, thank you, Marie.
All right, good morning, everyone. Thank you for being here. I'm really quite amazed at the crowd. You know it was club night last night, right? Some of you went. You should go next year if you didn't make it. Anyhow, so we're going to shift gears a little bit and talk more about task and task-driven behavior, and in particular the role of the executive network and the default network in goal-directed cognition. And this is going to be a different framing than you might be used to. I'm going to be discussing really the active role the default network plays in cognition when we're evoking a response from it, and how it can interact with executive control. So as we know, as Marie outlined early on, executive control involves flexibly adapting to changing circumstances and about integrating information over time. And it's thought to really play a big role in lateral prefrontal, lateral parietal brain regions. These are traditional classical frontal parietal control network. I'm going to be talking about the default network more from a functional perspective in its involvement in memory, and it's the ability of the hippocampus and lateral temporal structures, coding for episodic experiences, personal experiences, as well as the semantic representation of knowledge. And these are really drawing on, again, these medial temporal and also midline brain structures. So both of these processes, I believe, are actively interacting to guide both ongoing as well as future behavior, such that when we are engaged in executive control type tasks, we typically are performing operations on visuospatial information that's present in the environment. However, it's much more common, in fact, in day-to-day life that we're not simply working with stimuli that we have no prior information about. And oftentimes we're, in fact, using our executive control functions to draw upon what we know about the world and to adaptively apply them to our current environment. So in a way, I'm going to jump to the end, and I'm going to show you how I got here. I've performed a number of studies with Dan Schachter looking at this construct of autobiographical planning. And what you can see is when people are actively making plans for their own individual life in order to accomplish certain goals, you see a large-scale coactivation pattern between regions of the default network, including medial prefrontal cortex, posterior cingulate, medial temporal lobes, lateral parietal regions, as well as activation in lateral prefrontal cortex, lateral parietal regions, as well as dorsal anterior cingulate. So this suggests that both of these two networks, the frontal parietal control network and the default network, can be coactive and potentially coupled during an active cognitive task, during a personally relevant goal-directed cognition. So planning in general is considered a complex form of cognition related to goal-directed behavior. Some would consider it executive function, others not. But by and large, it's been studied with very simple radical reductionist approaches, such as the Tower of London task. And in this case, this task was introduced to Tim Challis, to the neuropsychological literature. It's been imaged substantially. And what we see is a stimulus apparatus with 3 bars, 3 discs. And the objective is to essentially acknowledge the goal position and the start position and move these discs in such a configuration as to match the sequence. So in this case, what we would do is move the green disc forward 1, the red disc forward 2, and then put the blue disc on top of the green disc, and then move the red one back again. So this is a moderately difficult planning task. And the objective, you can measure reaction time, you can measure accuracy in terms of the number of moves it takes to solve this puzzle most efficiently. So this is a very reductionist view of planning, but it also involves certain core elements. There's a recognition of a goal stage relevant to one's current starting point, and you need to integrate and inhibit information over time in order to accomplish that goal. So when we image this kind of task, what you see is a fairly robust activation of the dorsal attention network, presented here in gold colors, and a suppression of default network structures here in green. However, there was some tension in the literature for a number of years whereby if people imagined their own personal future, you see robust engagement of the default network, as well as in the medial temporal lobes, as well as these medial prefrontal, medial parietal areas. So it was unclear, really, if imagining oneself in the future is engaging the default network, yet planning for one's future, or planning in general, is engaging these goal structures, the dorsal attention, and these networks are anti-correlated. It was really unclear how one could actually ecologically plan for one's life. Like how are we actually transmitting information across these systems? So what we did is we devised a new task of autobiographical planning, and the idea was to draw upon real-world areas of concern from our subjects. So we prospectively sampled a large group of undergraduates, what their goals were, what they saw as certain steps in order to accomplish these goals, as well as some obstacles they need to overcome along the way. And we distilled down this stimulus set to the most reliable set of goals that these young adults had, and we basically structured the task to visually match the Tower of London. So just to give you an example of what some of these features are, we'd have a goal, which is freedom from debt. This is the most reliable goal across all of our undergraduate samples. And some steps in order to accomplish this goal: get a good job, save money, and an obstacle is they'd want to have fun. So what we did was we had a similar apparatus as the Tower of London. However, the goal state was projected at the base, and the discs were filled with these terms. And the objective for the subject was to integrate these steps and obstacle into a personally coherent plan in order to accomplish this goal. So if they were to think that they're in debt, but if they accomplish a job as an investment banker, they'd set aside a certain percentage of their income, and then with the rest of it, they could have fun. That'd be a way to accomplish this goal state. Now, you'll notice that they were in this planning stage for 15 seconds. Obviously, getting out of debt is something that you could think about and talk about for hours. So this is really just a snapshot of that moment where people are engaged in this task. And then they indicated how much detail through their plan. Subjects also performed the standard Tower of London task. as well as a baseline task where they counted vowels, which has been a baseline for both autobiographical memory as well as Tower of London type planning tasks. So when we decompose these data with partial least squares, what we see is there are two significant latent variables. The first one dissociates the autobiographical planning condition, depicted here in blue, from the visuospatial planning task, depicted here in red, with the counting task tending to co-vary along with visuospatial planning. And what I think you'll notice from this topology is this task-driven activation really nicely replicates the anti-correlation that you see between the dorsal attention and the default network systems. In the second latent variable, we saw that both planning tasks tended to co-vary together and were dissociated from the counting task. And what you'll see here is both autobiographical planning and visuospatial planning are engaged in these regions of the frontal parietal control network, including lateral prefrontal cortex, anterior insula, dorsal anterior cingulate, and so on. So reviewers being how they are, we also did a univariate approach to this, where we took a single estimate of the entire network and extracted bold signal. And what we see is that within the dorsal attention network, that autobiographical planning is suppressing activity here, and it's engaged during visuospatial planning. Autobiographical planning is engaging the default network, and that network is suppressed during Tower of London. However, both of these tasks are significantly engaging this extended frontal parietal control system that was estimated in resting state. So we're also very interested to see how these networks are potentially interacting in the course of a task. So we extracted the time series of these bold signal and correlated their activity. So what we found is that during the Tower of London task, or in autobiographical planning, the dorsal attention network is significantly coupled with the frontal parietal control network. That's on the y-axis. And the default network is decoupled from the frontal parietal control network during the Tower of London planning. However, when participants are engaged in autobiographical planning, the dorsal attention network is decoupled from the frontal parietal control system, and the default network is coupled with the frontal parietal control system. So what we see here is a significant task-by-network interaction where the frontal parietal control network can flexibly couple with either the default network or the dorsal attention network, depending upon the domain of the task. It'd be very nice to be able to examine how these regions are actually functionally interacting with each other in the context of task. We're working on methods now in my lab to do that. However, in the meantime, this is an estimation of how these regions within each network are coupled with each other just during the resting state. And what this suggests is that we have this frontal parietal control system depicted here in green. It shares extensive functional connectivity at rest with both the dorsal attention network in red as well as the default network. And what we can see here is that there is a rich functional neuroanatomy that allows for the interaction and integration of information between the default network as well as the frontal parietal control system. So just as an interim summary, we see that planning content is domain-specific, but the process of planning itself seems to rely upon these executive control network. And default network activity, when it's coupled with the control network, can support goal-directed cognition, goal-oriented cognition, which really stands in contrast to a lot of studies which have arrived upon this conclusion that the default network is antithetical to goal-directed cognition, and this serves to really correct that perspective. And really, it's this interposed control network can potentially integrate information across both domains, both within the default, from memory systems, as well as from the attention network. And this is really potentially a reconfiguration of memory in concert with cognitive control operations. And this serves to guide behavior. So I just showed you a fairly unconstrained task showing evoked default network response. We did due diligence in speaking with our subjects afterwards to make sure that they were, in fact, compliant with our task. We revisited each one of these goals. And in fact, we can see parametric modulation of the default network in accordance to some of these goals. But still, admittedly, it's fairly unconstrained. So when I established my laboratory at Cornell, I've been really trying to get a handle on the nature of the functions of the default network system. So let me just give you an example. So this is a human face, but you know more about this face. You know a lot. In fact, within 100 milliseconds, the brain response is different than you would be to a face that you've never seen before. And this really, I think, serves to highlight the spontaneous nature of the default network and its evoked response. It's really based a lot on the familiarity of signal. So you've been walking around this conference center for the last few days. You're in the poster gallery. You see someone and they smile at you with a a knowing look of familiarity. And it is extremely adaptive to have a very rapid response where you show that you know this person, to actually bring information to mind, such as their name, the nature of your relationship, and it provides you a context by which you can navigate that social space. And if you can't recognize the face directly, maybe you saccade down to look at their name tag, in a way foraging for information to help guide your behavior in this unstructured environment. So our question is, can we leverage this kind of default network process in order to facilitate executive control? And we did so in the context of an NBAC task. Most of you will know this is when you see a stream of stimuli and need to make a button press response if the stimulus from N ago is the same as what you're currently viewing. And typically, this kind of task leverages either numbers or letters or even in some cases shapes that you can't put an easy name to. And the objective is to provide our participants with information to perform executive control operations on that are devoid of any sort of personal meaning. And there's an obvious reason for that, is because people are coming to these experiments if there's known information is potentially a confounding variable. However, we wanted to turn this around and try to leverage that. And we did so with a faces NBAC task, where participants were shown a face for 500 milliseconds followed by a 2-second delay. And in this stream of faces, there were different matching contexts. So on the one hand, we just had people matching to anonymous faces. In another, they'd be matching anonymous faces with a famous face interleaved long-term memory probe distractor. And then in other cases, the FAF triplets, where they're matching famous face interleaved with an anonymous face. And here what we hypothesized that we would have better performance when we're matching upon long-term memory, because long-term memories can scaffold and support executive control functions, and further, that this would be supported by the default network. So first, what we found was indeed a facilitation effect whereby participants were both more accurate and responded faster when there are matching famous faces relative to matching anonymous faces. So first off, what we found was overall this NBAC task nicely engaged these regions we've long associated with executive control. And if you look at the ventral surface, you additionally see activity in the putative fusiform face area. So this is just a very nice proof of concept where you see these working memory regions, and it's a working memory for faces task. So when we look at the individual events, for the first time that someone sees a famous face relative to the first time they see an anonymous face, what we see is famous faces are engaging these midline structures we associate with the default network. We see some ventral posterior cingulate, retrosplenial cortex, ventral medial prefrontal cortex, the hippocampus, as well as perihippocampoid regions, lateral temporal areas, as well as some areas outside the default network, including lateral prefrontal and the anterior insula. What's interesting here is that the anonymous faces, in contrast, seems to be engaging this extended frontal parietal control system, suggesting that those areas are potentially downregulated when you have an increased activity in the default when perceiving a famous face. And if we look over the interval of the face match going across the full triplet, what we see here, again, is sustained activity within these default network structures in ventral posterior cingulate, retrosplenial cortex, ventral medial prefrontal cortex, and many others associated with the default network. So we have here is default network and executive network interactivity involved in goal-directed cognition. They can both interact and couple, or they can preferentially trade off. And I really think this provides some provisional evidence, at least, that how working memory can gain access to long-term memory and representational knowledge. And this serves to highlight some of the features of the default network and how it can be actively engaged in cognition. So what I would like to plead with many of you today is to let's move away from this idea that this is a task-negative network. I don't think that really respects the role of cortex and its core functions. We wouldn't necessarily discount lateral prefrontal cortex. It doesn't make sense to me anymore to say that there's a task-negative or task-positive network. We have this integrated cortical systems that are all actively engaged in cognition. So with that, I'd like to thank you and to acknowledge my lab. All right.
Is my mic working? So I'd like to thank all the speakers, and we've got a couple minutes left. So if people have questions, please come up to the mic. We'd be happy to answer them. Or maybe people haven't had enough coffee. So any questions, comments? Sure, great, thank you.
I have a question for Dr. Morton, and he said that the graph theory metrics are less reliable, so let's go to dynamic. Did you also contrast in your study, within the same study, measures of graph theory and the dynamic? That would be interesting to me, especially given the very first introductory talk where the graph theory metrics were actually quite interesting. So the comparison between the two, did you do that back to back?
I haven't, no. And it's a very good question for sure. And I think what I mean simply by the graph metrics being unreliable is that the correlations upon which any graphical characterization of network organization are based are going to vary profoundly within the measurement period, right? So this is work by Chris Honey. So even if you expand the measurement period to 10 minutes and you compare 10 minutes of resting state with 10 minutes of resting state, the test-retest reliability of any coupling is going to be in the neighborhood of around 0.4 or 0.5, which is not terrific, right? So if you build a graph from couplings where the test-retest reliability is in the neighborhood of 0.4, 0.5, you could only imagine that the graph metrics themselves are also going to be subject to terrific unreliability. And we just simply say, well, that's because the spatial features of these networks dynamically vary over the measurement period. As to whether or not the dynamic metrics themselves have better test-retest reliability, I think is something that the jury is still out on, and I think it's a really important question. I know that Matthew Hutchison is working on some of this stuff. Lucina actually has some interesting, there's a sub-story in the doctoral student Jason's work that speaks to some of these issues of reliability. So I'm not going to say now that dynamics solves the reliability problem. What I will say, though, is that graph metrics suffer from problems of test-retest reliability.
Yeah, definitely. Thank you very much.
Can I follow up on that? I mean, I think the other thing is when I presented my results, I didn't really focus on what the metric was for one particular node. Mm-hmm. Rather, my takeaway message was the general pattern across the nodes, that we were consistently seeing that those individuals with higher EF were showing that the characteristics of nodes within the frontal parietal network seem to show less integration. So that's also one of the reasons why I presented the data that way, than saying, ooh, let's look at the DLPFC node and talk specifically about that. Any other questions? Yeah.
Thank you. I want to know the executive function is served by a frontal parietal network. How do you think about whether these brain regions exert brain executive function by sequentially or parallelly? Thank you.
Okay.
You've been elected.
Oh, no. Okay. Do you have anything to say? All right. Sequentially or parallelly? Well, I think none of us have actually shown data today that would address that. But I mean, from some of our work in using things like greater causal analysis and other causal modeling techniques, which have their own inherent limitations, we have seen some evidence that some of these nodes tend to sort of precede activity in other nodes. So in our own work, we often see things like the dorsal anterior insula activity can be used to predict what's going to happen in DLPFC and anterior cingulate, for example. But I mean, I do realize that it's always, I'm sure there was a symposium on skeptical connectivity. There was probably some other symposium on like don't do Granger a few years ago. So, I mean, there's always going to be some limitations with trying to infer timing and causal sequences from fMRI data. But But I think, I mean, from what we can tell, there do seem to be some nodes that precede others. I don't know if anyone else.
The other thing I would say is I agree that that's not well addressed by fMRI data. For example, some of the work that we've done in conjunction with Wendy Heller and Greg Miller, we've looked at the relationship between DLPFC and anterior cingulate. So we've argued that DLPFC is essentially in some ways going to be maintaining task goals, and that the anterior cingulate is more response related. It's sort of cleaning up at the end. If you don't have good top-down control from DLPFC, ACC kicks in towards the end because somebody's got to do something before you emit a response. And as I said, in conjunction with Wendy Heller and Greg Miller, what we've done is essentially look at the sequence of activity between dorsolateral prefrontal cortex and ACC using essentially fMRI seed-based dipoles. And that is very interesting in that it gives you some of this information. So, for example, when we look at people who have do well like on a Stroop-like task, what we find is that they show high DLPFC activity in an early window, and essentially ACC doesn't predict their behavior. So we're looking at this sequence to predict behavior. Okay? We find, on the other hand, that if you have not so good DLPFC activity, so in our model, not so good top-down control, that if then later on the ACC kicks in, what you see is that people have poor or elongated reaction time. Okay? However, if they have poor DLPFC activity followed by poor ACC activity, then they show more errors. Okay? So the idea here is that there may be some temporal sequences in some cases. As I said, sort of the anterior cingulate is playing cleanup. When it does, you have elongated reaction time if your DLPFC isn't working. If it doesn't play cleanup, then you make a high level of errors. So there are some models of that nature out there, but I think that EEG and MEG are probably much more well suited to looking at those issues than fMRI in terms of timing. Okay.
Yes, I wanted to ask the second speaker, Miss Dr. Uddin, if you're aware that during your task, I mean the erotic one, maybe some of your subject, at least someone, they're not doing the task, but they're, seeing erotic images. And another question is about the wonderful matrices that you extract from the temporal windows. What do you mean with the brain state in a context in which you don't know what the subject is doing because it's a resting state, so-called the resting state, so you don't have any information about the behavioral state of the subject.
Mm-hmm. So the first question, there was eye tracking data collected, so you could see subjects that were, there are a few who didn't perform the task at all, were excluded from analyses, so, but most of them were doing the task with varying degrees of accuracy and reaction time. For brain states in the resting state, that's a very good question. So I tend to think of it as the propensity for the intrinsic connectivity to be in a particular configuration or other. So in most resting state scans, they're usually 5 or 10 minutes, but in this particular Human Connectome Project data set, there were 4 15-minute resting state scans. So you've got sort of more of the full range of every possible, I think, configuration that a brain could possibly be in. And so it may not be and probably isn't linked to sort of a moment-to-moment cognition, but rather just the sort of overall, I think, the question you're asking is really like, what does resting state mean or measure, and what does it mean for behavior? And I think that's a bigger question that I don't want to even try to tackle. But I think that, in general, the brain tends to occupy certain patterns, and those states are what we see as coming out, some states more frequent than others in these long resting periods.
Okay. Great. Thank you very much for coming. We really appreciate it.